So let's talk first about what are the two types of tests. So when I talk about test, I have either a cytological test or a DNA test. So if I am studying the cell as a whole, it is called a cytological test. Now, these cytological tests requires a geneticist. They require an expert who can actually look into the cell, look into the nucleus, look into the chromosomes. Now, the problem with the cytological test is there is a limit. I can only maybe count the number of chromosomes, see the bands, arrange the chromosome in its specific order. But if I want to look at details of the chromosome, what is the pattern, what is the banding, it might not be possible just with looking at a microscope. So after cytological test, the better test, which are called as DNA test. Now these DNA tests, what they do, they fragment the chromosome. That is, they make an array out of it, right? And these arrays, they can be read by a computer. So cytological tests, they are very good, but they are limited to the cell. They are limited to the counting of the chromosome, and they need a human. Now, human can only do things till a certain level. Therefore, DNA tests came into progress. Now, this DNA test, they will lyse the chromosome, they will make it into single strand, they'll compare it with the normal, and they will tell if the gene is normal or not. So the basis of genetic disease was cytology. And then once the AI started, once the computerized era started, the DNA test came into play. So the first thing we will discuss about is cytological test. Now, the first test in cytology is FISH. What is FISH? It is fluorescence in situ hybridization. Now, in this FISH, what do they do? It is again a microscopic based test. You need to see in microscope what is happening. They take a cell, they take out the chromosome from it. This chromosome they denature, and then they take out single stranded DNA from it. This DNA they hybridize, and they hybridize it with a fluorescent label, another DNA, which we have as a proband. Now, this fluorescence is then studied in the microscope. Now, once you have a fluorescence, it will get attached to the DNA. Now, how do the report comes? We are not worried about the type. How do they do the test? We are worried about the implication. Now, how do we get the report is, so they had the chromosome, they had probes. They have attached the probes and they have color coded the probes. So 13 is seen by green, 18 is seen by aqua or blue, and 21 is seen by orange. Sex chromosome also they will see, but they will not report because according to PCB entity, sex cannot be revealed. So how many signals they see that they will report? So green, they are seeing 2 signals, that is euploid. Blue, they are seeing 2 signals, that is again euploid. Orange, they are seeing 2 signals, that is again euploid. Sex chromosome, they are seeing 2, that is again euploid. So normal diploid, normal diploid, normal diploid, normal diploid. Here you can see 2 green, 2 red, 2 aqua. This one they will not show. So this is how a normal report of FISH comes. Now FISH is an excellent test to give out our aneuploidy results. Now generally we have limited it to 5 proband, 5 nucleus, 5 probes. You can even do it for many probes. But the problem in many probes is like we are analyzing a particular number of cells. For example, if I'm analyzing 50 cells, it will tell me about the 50 cells. If there is mosaicism in 51st cell, it will not tell. So it has a limitation. It is a very good test, but it is limited to certain number of cells. So FISH can tell you aneuploidy. FISH can even tell you mosaicism. Now it depends. If I'm counting 500 cells, the rate of me catching mosaicism is better. If I'm checking only 50 cells, the rate is less. So it depends how much tests we are doing in how many cells and till what extent we have to go. Now, if my doubt is more, I can always call up the lab and tell, I am strongly doubting aneuploidy. Please keep a check of more cells. Usually they do around 100 cells. You can always ask them to do 200 cells. They can always ask them to do more. So this is about FISH. Now, in case we have an abnormal report, then how does FISH report? So this was a patient. high risk for trisomy in NIPT. She was also 38 years old. So her sample was sent for fish and it came out to be abnormal. So they had 2 of green, that is 13, 2 of aqua, that is 18, but 3 of orange, that is 21. So this excellent test reported that yes, your high risk NIPT has proved to be actually aneuploidy. Now, if they have read 50 cells and they have seen aneuploidy in all of them, they will report 100% aneuploidy seen. If they see aneuploidy in maybe half of the cells, then they will report mosaic pattern, half normal, 50% normal, 50% mosaic. Usually we say that if mosaicism level is less than 30%, it is not actually relevant. That means the patient will not show signs of mosaicism. Got it? Now, if at all you have a sex chromosome aneuploidy and it comes in your fish report, then the labs report it, because in PCP entity you cannot report the gender, but this is a mismatched gender. So green normal, aqua normal, orange normal, but sex chromosome, they had trisomy. So here you can see 2 red and 1 green. So this actually was XYY, that is super male. Understood? So it is a good test for aneuploidies, fish, cytological test done by humans. Now, the second cytological test, which is a complement to fish and is a very good test, still used very widely, is a karyotype. Now, all of us know about karyotype. So this is shown in blood. We can also do an amniotic fluid. You just add colchicine, you burst the cell, you fix them, you put a stain, and you analyze the metaphase of the cells. Now, once you analyze, you actually need a human to arrange the cells in the specific place. Chromosome 1 ka identify you have to do, and you have to place it there. Chromosome 2. Nowadays, computers help us in identifying the type of chromosome, match it and place. But still, we have to, the geneticist has to sit and they have to do it. So this also takes time. This also takes expertise. This also takes microscopic analysis. And this is also a cytological test because you're actually bursting the cell, taking out and counting it. There's no computer involved in hybridization or PCR. So this is how the karyotype report comes. Now chromosome 1, see, you can see the long chromosome, you can see the short chromosome. You have one person has to sit, they'll get it like this, and then they have to sit and they have to do this. So karyotype will also complement your fish test. It will tell us that yes, there is no aneuploidy. Now, if at all you have an abnormal karyotype. So abnormality in karyotype can also be a translocation. Now, you have to understand that translocation is such a disease. That means the one, so in this case, you have a translocation. What translocation you have? You have translocation between the long arm of chromosome 1 and long arm of chromosome 2. So there is some genetic material which has come from chromosome 1 and it has gone to chromosome number 2. Now, this pattern is only being recognized in karyotype. If at all you go to a PCR, PCR में क्या होता है? They will, so for example, microarray needs a PCR testing. What they will do? They will fragment the chromosome. They'll get the DNA. They will complement it with the other DNA and they will print कि हाँ भाई ये वाला complete. Now translocation में there's nothing loss or there's nothing gain. So in PCR or in microarray, the report will come as normal because there has been no loss, no gain. Jitna chahiye tha utna hai. So only a karyotype, that is a human, can tell that this was supposed to be here and it has gone somewhere else. The puzzle has been mismatched, but it is complete. Balanced translocation does not cause any problem in that human per se. मुझे कोई प्रॉब्लम नहीं होगी अगर मुझ में बैलेंस ट्रांसलोकेशन है, बिकॉज इन टोटल आई हैव ऑल द जेनेटिक मटेरियल विच आई नीड. Understood? So karyotype is the only test which can tell you about translocation, and balanced translocation is not an issue. Balanced translocation means balanced hai, human is complete. So this is one more example of a karyotype which is positive for chromosome number 21. Now here you can see this is a pure chromosome 21. What is a pure chromosome 21? In which you have one additional chromosome 21. It is not going and sitting on somebody other chromosomes back. So this type of chromosome is called as pure. This is the most common type of T21. That is 95% of trisomy 21 is pure chromosome, and it usually does not tend to reoccur. But definitely you need karyotype to know which type of chromosome it is. Your fish will not tell you which type of Downs it is, but your karyotype will. So that's why अगर fish में आपका positive आ गया, you need to complement that test with karyotype so that we know कि ये Robertsonian translocation नहीं है, it is not an isochromosome and it is a pure Downs, so that for the next pregnancy we are aware. In these cases जिसमें ट्राइजोमी प्योर टाइप की आई है, for the next pregnancy you need not do an invasive, you can also do an NIPT for screening accordingly. Invasive is definitely best, but NIPT is also legit in these type of cases because the recurrence rate is less. But अगर रॉबर्ट्सोनियन आ गया या फिर आइसोक्रोमोसोम आ गया, in those cases you need to do an invasive testing. So this also is a karyotype. The case which we saw in which there was XYY, it is the same patient's karyotype. This is single X and YY. So in these cases also, if genetic XY is abnormal, the PCP-NDT rules are not valid here and the labs will report, because this is the problem which is been diagnosed in the fetal life. So here also they can make a decision to terminate if at all they want to. Here, this is the translocation wala. Translocation from chromosome 1 to chromosome 2. Now, see how minute it is. है ना? And it is just translocated from here to here. So, how do you write this translocation? 46 chromosome complement is there. There is translocation from chromosome 1 to 2 in this area. 1 ka 23, 2 ka Q ka 21. Q is long arm. So, long arm ka 23 locus has gone to long arm of 21 locus of chromosome number 2. But it is complete. Now, this thing can only be done by humans. The computer will not know. Computer will tell ki everything is there, everything is normal. So, in cases of recurrent pregnancy loss, where balanced translocation in one of the parent is a cause, microarray should not be done. Parental karyotype is necessary. Understood? To rule out balanced translocation. Now, what does balanced translocation lead to? That I'll tell you in future slides. Okay, now we start with our DNA tests. So the first DNA test is QFPCR. Now, what is this QFPCR? It will take the cell, it will lyse the cell and it will amplify it. Now, after amplifying, it will tell you how many alleles are present. So, for example, if it is a normal allele, it has amplified the cell and it has seen two of the amplification probes are there. So, disomy, normal allele ratio 1:1. Now, it is comparing with the it has a normal proband and it is comparing with your sample. So your sample comparison, normal proband, normal ratio is there 1:1. Now, if it is trisomic, now it has 2 here, comparison to 1. Got it? So 2:1 का ratio आया है. Now, for example, trisomy 21 है. So 2 in your sample and 1 in the proband. Now, if it is trisomic, that is triploidy. So it is coming to मतलब 2 sets compared to 1. If it is uninformative, that means sometime contamination आ गया या monosomy है, तो it cannot distinguish. It cannot distinguish क्योंकि उसके पास एक ही बैंड आ रहा है ना. Either it is a pro band का प्रॉब्लम, दैट मीन्स उसका जो बेसिक था उसके पास जो कंपैरिजन के लिए उसका प्रो बैंड था वो प्रॉब्लम है, या फिर सैंपल में प्रॉब्लम है, या फिर इट इज मोनोसोमी, या फिर द टेस्ट इज नॉट एबल टू प्रोसेस. सो इन दीज केसेस इट इज बेटर टू डू अ साइटोलॉजिकल टेस्ट, दैट इज फिश, जिसमें इंफॉर्मेशन नहीं आ रही है. Now this is how a QFPCR report comes. It is chromosome 21, 18, 13. It will check कि diallele था कि disomic था कि नहीं. If all the things are normal, it will give you a normal study. Now QFPCR and fish are complementary. कैसे complementary है? So both of them will tell about common aneuploidies. Both of them can give you a report in 48 hours. But ये जो है इट विल कैलकुलेट, फिश जो है इट विल कैलकुलेट अ पर्टिकुलर नंबर ऑफ सेल्स. सो इफ आई नो कि 50 सेल्स मैंने दिया है, बट QFPCR कैन डू योर टेस्ट इवन इन वन सेल. But the problem is, if it is doing it in one cell, it is seeing only one cell. Now, if it is seeing only one cell, it will not tell you about mosaic at all. So, mosaicism is only caught nicely in fish. Now, I can increase my QFPCR threshold, but then usually they don't take up many much cells and they'll get confused. The computer gets confused. The computer को इतना पता है कि अगर मेरे को तीन वो देखने हैं और मेरे को तीन दिख रहा है, इट विल रिपोर्ट एज नॉर्मल. इट कैन नॉट टेल यू कि इतने सेल्स में नॉर्मल दिखा था, इतने में नहीं दिखा था. दैट यू कैन डू ओनली बाय माइक्रोस्कोप. ओके. Now, next thing is microarray, as I was telling you. So what do we do in microarray is, you have a healthy cell and you have an aneuploidic cell. Now, this healthy cell and aneuploidic cell will culture and we will take out the RNA out of it. After taking the RNA, we will put it to primers, उसके complementary base pairs. So for example, ये healthy cell का मेरे को RNA मिल गया है. Now I am attaching this RNA to my probe, which is the normal human का probe. Now, if at all everything is attached, it will give you a green signal. If at all something is extra or it is not attached or it is, so for this, these are the normal, इधर deletion है, so it will not be attached. So this will light up as red. This will light up as green because this has fluorescence. So after that, after everything, we have a microarray chip or a microarray plate. इसमें pits रहते हैं. Alright, so in these pits, they will attach all the DNA here. And because they have multiple DNAs, they will attach it to multiple probes in the plate. Now, these plates will give you a result. In the end, the computer will make you a chart and it will give you a result. It will tell कि इतने सेक्शन में हमारा हाई फ्रीक्वेंसी था, इतने सेक्शन में हमारा लो फ्रीक्वेंसी था. एंड दीज चार्ट दे विल मैच टू नॉर्मल ह्यूमन. अगर कहीं भी लो फ्रीक्वेंसी आए मतलब the probes were less, that means there can be a deletion. How do you know high or low? It depends on how fluorescent it is. जितने ज्यादा डिलीशन होंगे उतना कम फ्लोरेसेंस यहाँ पे चमकेगा, जितने ज्यादा डुप्लीकेशन होंगे उतना ज्यादा फ्लोरेसेंस रहेगा. सो अकॉर्डिंगली दिस चार्ट विल टेल यू कहीं डिलीशन है कि कहीं डुप्लीकेशन है. बट इट कैन नॉट टेल यू ट्रांसलोकेशन, क्योंकि अगर मैं डाल रही हूँ वो लाइज होगा तो मुझे नहीं पता कहाँ का कहाँ गया है. वो लाइज हो गया एक बार तो उसके बाद इट कैन नॉट टेल यू इफ इट हैज ट्रांसलोकेटेड फ्रॉम वन प्लेस टू अनदर. सो माइक्रोएरे इज अ वेरी गुड टेस्ट. इट कैन नॉट टेल यू ट्रांसलोकेशन, इट कैन टेल यू एन्यूप्लॉइडी बिकॉज वो बहुत ज्यादा चमक जाएगा, and it can tell you deletion and duplication which karyotype cannot tell. So what are the types of genetic mutations? We have seen here substitution, that is one entire area is substituted by other. Deletion, one part is missing. Insertion, one part is extra. And inversion. So microarray can tell you substitution, microarray can tell you deletion, microarray can tell you insertion, but it cannot tell you inversion. Inversion and translocation same to same. मतलब inversion is just inverting there itself. Translocation is going from one chromosome to another. So these inversions and translocations microarray cannot tell. Now generally inversions and translocation do not cause a problem in fetal life or adult life. It will cause a problem later when the individual wants to conceive who has the translocation. So this is how a microarray report comes. Now a microarray, see if you can see, it has an array. है ना, इट विल टेल यू कि गेन है कि लॉस है कि लॉस ऑफ होमोजाइगोसिटी है. You don't have to worry about this. If at all there is no gain or loss, they will give us normal chromosomes. If at all there is gain or loss, they will tell इधर ब्लू आ जाएगा इधर रेड आ जाएगा, गेन है कि लॉस है. दैट मीन्स डिलीशन है कि डुप्लीकेशन है. नाउ टू अंडरस्टैंड माइक्रो एरे, ये समझो कि आप एक सुपरमार्केट में जा रहे हो. नाउ इफ एट ऑल देयर इज समथिंग मिसिंग, you will know. You are the analyzer for micro, you are the computer. You will know कि something is missing, है ना. कहीं अगर overcrowded है, you will know that things are extra here. But for example, if I keep these things here and these things here, you are not worried. You will know कि हाँ जितना मुझे सामान चाहिए उतना है, तो you will not report to your senior. If deletion is there, कुछ नहीं है, you will report. If overcrowding is there, you will report that this is not clean. But if things are just kept from here to here, you will not report because it is there. So translocation you will not report, deletion you will report, duplication you will report. So as a microarray, translocation it doesn't report, deletion and duplication it reports. Now, does an abnormal microarray report look? So this was one patient in which I was suspecting trisomy 13 because all of the features were trisomy 13. So it had echogenic foci, bilateral orbits were fused, that is hypotelorism. The cranium was thin, it had cystic spaces, mentally retarded holoprosencephaly, proboscis was there. Everything was suspicious of trisomy 13. So I had sent for karyotyping. Now the culture failed and they could not do a karyotype. The good thing about DNA tests is even if the culture fails, है ना, you can still extract some DNA. एक cell भी मिल गया तो हम microarray कर लेंगे. But if the culture fails and the cell do not grow, we will not get metaphase. So you cannot do a karyotype. So in these cases, the culture failed for this amniotic fluid, and they went ahead and they did the microarray. Now what they got is there was duplication in chromosome number 13 and deletion in chromosome number 18. So they reported this as abnormal. राइट सो व्हाट हैपेंड, जहां जैसा मुझे लग रहा कि 13 में प्रॉब्लम है, 13 में डुप्लीकेशन आया बट देयर वाज अ डिलीशन इन 18. नाउ अगर कहीं से डिलीट होके कहीं से डुप्लीकेट हो रहा है तो इट इज अ ट्रांसलोकेशन. बट व्हाई वाज माय माइक्रोएरे एबल टू कैच इट? बिकॉज जो हमारा डुप्लीकेशन था दैट वाज 35 MB. दैट इज मोर देन हाउ मच वाज डिलीटेड. डिलीशन सिर्फ 7 MB का था, डुप्लीकेशन वाज 35 MB. So that's why the microarray was able to catch. And that's why they reported it as abnormal. Now, when you have this, थोड़ा सा एक जगह duplication कम है और एक जगह deletion ज़्यादा है, what do you suspect? You suspect कि it might be something called as unbalanced translocation. क्योंकि एक जगह से दूसरी जगह जा रहा है पर balance नहीं है. So in this case, then after receiving this report, I thought it will be very worthwhile to check the parents ki are they carrier for a balanced translocation which has become unbalanced in the fetus. So now this was unbalanced. Then I sent the parents ka karyotype. To my not so surprise, I got translocation in the mother. And what translocation? Exact 13 and 18. So here you can see they have marked it in red. If I don't know if you can see, this is 13. Okay, and this is 18. So because there was a translocation between 13 and 18 chromosome balanced in the mother was symptomless and the baby had features of trisomy 18. So these condition in which aneuploidy is there, you have to check for parents as well. Now, this was the father. Father was normal. So this was father was 46XX, but the mother came out to be translocation 13 and 18. Now, next test is Sanger sequence. Now, this Sanger sequence is nothing but they take a strand of a particular DNA in which one gene you can check. Now, usually they take only the strand in which our interest gene is there. So, for example, I have to check this particular gene which causes thalassemia. So what I'll do? I will do Sanger sequence for thalassemia. Now I know thalassemia gene is located in chromosome number 1. I will check only chromosome number 1 ka that loci in which this gene is present. Why to waste time in checking everything? Or why to overdiagnose? I want to check in peripheral venous blood for this particular patient who has got an HPLC positive. They want to check it because they want to see key future implication, the fetus is there or not, first thing. Second thing, it also confirms your HPLC. Now, it is also mandatory to check the parents before checking the fetus for invasive procedure because in sometimes HPLC can be wrong and some mismatch. So thalassemia, okay. But if thalassemia and HBD is there, you will not check the fetus because the clinical implications are very minor. So this is also a confirmatory कि HPLC का आपको confirm हो जाए. Secondly, you will know if the fetus actually needs an invasive testing or not. So Sanger's may you can check one particular gene if it is affected. Now Sanger's was one of the first test जो single gene को target किया. And when we came to know, Sanger's was started in 1970s. So when Sanger's started, people started doing Sanger's for multiple genes. So for example, अभी तो मुझे sickle cell पता है. I know that my patient is suffering from sickle cell. Now, in case I don't know the disease and I can see the symptoms, but I don't have any other tests. So in 1970s, people started doing Sanger's for 111111 gene, but that took around months to give the answer. But we don't have months. So people started doing prenatal testing. Yeah, preconception testing. We will take months together, we will check the parents, and then we will do for the kids. But that also was not cost effective or time effective, or ergonomics it was not good. So after Sanger sequencing, which took so much of time, then came out whole exome sequencing. Now this whole exome sequencing, it is done by NGS. NGS is also called as next generation sequencing. So after Sanger's came NGS. Now this NGS is similar to microarray, in which they take entire strand of gene, they compare it with normal, and wherever there is mutation, it will light up. So this whole exome sequencing, it takes very less time. It only takes around 2 weeks to 3 weeks at max, and 3 weeks we'll get reports. So the process only take 2 weeks, and it is able to massive parallel sequencing, we call it. That is massive reading of gene at simultaneous time, in the simultaneous time. So simultaneously it will read massive genes, and your time is efficient. You can read multiple genes together. Moreover, you can detect it during the conception period. So it saves the time also. So whole exome sequencing will not give you one answer, it can give you multiple answers. Sanger sequencing targets only one disease at a time, which is caused by a single disease. If a disease is caused by two genes, you cannot use Sanger's. You need to either do two Sanger's or you need to do a whole exome sequencing. Understood? So if I know the disease, I know it is caused by, so for example, cystic fibrosis. I know that cystic fibrosis is caused by CFTR gene. I know that the parents are affected. I know that the previous baby was affected by cystic fibrosis. I'll not waste time in detecting everything else. If my ultrasound is normal, I will just do amniocentesis and send for Sanger sequencing. Sanger sequencing for a single gene will give you a report in 1 week. Now, if, for example, the parents are normal, and my baby, my fetus shows ultrasound with multiple things, and I'm suspecting some syndrome. I don't know, because this is the first baby. I don't know if the baby will have some syndrome, and is it going to repeat? I don't know anything. So in those cases, I will send for whole exome sequencing. Now this whole exome sequencing will run the entire exome, and it will tell me kitne genes, how many genes are there, if they are mutated, if they are deleted, if they are duplicated, everything it will tell me. Now this is how a whole exome report comes. So what, see here, sorry for the patient name. This was a consanguineous marriage. It showed unilateral congenital talipes or clubfoot. Microarray analysis was done and it was normal. Microarray will just tell you deletion, duplication. It will not tell you single gene. So now it has to be evaluated further. Now whole exome will check single genes, right? So now this whole exome was run and we found a gene which was homozygous, autosomal recessive inheritance, and this had a Niemeline myopathy 8, which is autosomal recessive in condition, which is likely pathogenic. So now that I know that this baby has this CTB ka this cause, now I can go ahead, I can study about this disease, and I can tell the parents ki CTB ke alawa, if at all some implication more is there for this disease in this fetus. Moreover, after they want to continue, not continue, after this pregnancy is over, we can check the parents ki aaya kahan se. Thirdly, we can check it in the next pregnancy in a CVS sample. So many things you can do just by knowing the implication of that particular gene. So you have NGS, you have Sanger's. Sanger is reading one gene, NGS is reading multiple genes. NGS will give you more information, but more information is always not good information. So there are 5 categories in which it can give you disease information. First is pathogenic. That means this is the gene, it causes this disease, and this gene is pathogenic. Second is likely pathogenic. This is the gene, it is most probably going to cause this disease. Next is benign. This is the gene, unlikely it is going to cause any problem. Confirmatory benign. This is the gene, it will not cause a problem. Last is what we are very scared of is VUS. That is variance of uncertain significance. So till date, we have started doing NGS very recently. We still don't know, so many genes are there and they are being discovered on a daily basis. Now, if I have a particular gene which has still not been studied, and I don't know if this is going to cause some problem in the future or not, that is called as a VUS. VUS means variance of uncertain significance. Now, once, for example, I have done amniocentesis for a 20-weeker lady, in which in ultrasound I was seeing renal pelvic dilatation. Now I have done amnio, I sent for whole exome because the patient was like, Madam, I don't want to take any risk. What if some kidney disease comes in the future? I don't want to take any risk. You completely analyze everything. I said, okay, I will analyze. Now I did the amnio and it came out ki ABCD gene is there and it is a VUS. That means we don't know if it is going to cause problem or not. Now, if the lab only doesn't know, how will I know? So what is the implication of VUS is you need to classify it as benign or pathogenic. How will I classify? I have to see if this fetus, when it grows up, it will have the problem or not. So for this patient, it became null, 0 point nothing, no point of doing the amniocentesis. And I am not able to give her the answer also. So if at all a VUS comes, what is the next best step is, now I will check the same gene in the parents. Now, if in parents also it comes, parents are absolutely normal, so then I can classify the gene as benign. But if parents also it doesn't come, I still don't know if it is pathogenic or benign. But according to the laws, I cannot abort the pregnancy. I cannot make a decision on the basis of VUS. So if you get a VUS that is unknown significance, you check the parents and hope that it comes out in the parents. Because if it comes out, well and good, you can leave the baby alone, you can leave the parents alone, continue the pregnancy. But if it doesn't come out in parents, again you are at 0 point. So VUS is something which is being classified daily, but you need a long time to classify it. The more we are going forward, the more we are classifying it, but then the more VUSs are coming. That's why till very recently, whole exome sequencing without any previous history was not being done in fetuses. But we were missing on so many diseases, so now they are doing again, and we are somehow trying to deal with VUS.